Great. Okay, so uh, we have been looking at these simple models, and then I've been talking about how to make those simple models a little bit more realistic, and what are the various conditions that are not really there in the models, and therefore it becomes difficult to use them directly, and so on and so forth. So there were two kinds of questions that I got asked. One was that if the simple models are so simple, then and to the point of being not useful, then why should one even bother with them? Okay, and that's a very valid question. The point is that they are not as not useful as they looked from my presentation. So I believe that there is something I need to say in their defense. The second point is that some people ask that we said that a mechanistic model is one that is giving us some understanding about how the system is working. However, it's pretty clear that we aren't getting too much of understanding from these simple models. So then what is the mechanistic model that is being talked about? Okay, where are those mechanistic models? What is the nature of those models? So in order to answer these two questions, I'll have to do certain topics which I normally don't take as part of the second year class. I mean, I don't take them at ICER at all. But since you guys have asked this question, and since this particular course at ICER Pune allows me to be a little flexible, what I'll do is I will end up introducing a short digression. Okay, it's still in the context of the models, it's still very much relevant to what we have been discussing, but it is something that is typically not a part of an introductory course. Okay, so what I'll do is I am going to take some examples from the kind of work that I have been doing both for my doctoral work as well as in here in my lab for the last several years, and I'm going to use that as an example to answer some of these questions that you guys have asked. By no means this is the only way that simple models can be used. By no means this is the only way in which mechanistic modeling can be done. But these are just examples from the literature. And the reason I'm using them is because I'm very familiar with them. That's about it. Okay. So in the context of whatever I'm going to discuss, the mathematics part of it is again not very important as far as this course is concerned. Okay, it's not that they're not important by themselves, of course they are, but not in the context of this course. And therefore, in whatever I'm going to talk to you about, what you guys should focus on is the approach that we are taking to solve certain issues. It's not about the precise form of the equation or the precise form of the theorems that are being used over there. If you guys are interested, let me know. I will guide you towards the relevant literature. All the references are anyway given in the presentation. But again, I emphasize the maths is not the key over here. The biology is. Okay. So with that little introduction, we'll go in for the presentation. Okay. Great. Great. So can everybody see my screen
Yes,
over
sir.
here?
Yes, sir.
Okay. Okay. Great. Great. So I'll be talking in the context of stabilizing biological populations, which has been a major theme in my lab for last quite some time. And as you can see over here, that I'm explicitly talking about the experimental biologist's perspective. Okay, so if you are on the bench, if you are handling organisms on your own, then how will you go about seeing population dynamics from the perspective of the models? So it's a bringing together the two sides. So controlling the dynamics of real biological populations is, and when I say real, I mean actual organisms as opposed to models. Okay, Now, this is a very venerable topic, and it starts almost as soon as controlling chaos control starts, which is with a seminal paper in 1980 by O.G. Y. Ott, Reboghi, and Yorke. Uh, the problem is that although a lot of work has been done in this context, most of it has been done not from an experimental perspective, but mostly from a theoretical perspective. Now, when you take all those theory and you try to apply it, there are several issues that you get into. A, most of the theory assumes that you know what is the model that is governing the dynamics of the system, but in reality, you really don't have a clue. Okay. Most of the things, most of the methods that are proposed in the chaos control literature, most of them actually end up perturbing the parameters of the model. So in the context of the Ricker model that we studied, they are about some perturbation to say the R, or some perturbation to K, and so on. Now, much of the time when you are dealing with a real system, you don't have an experimental handle on these things. Okay, so R is not something that you can directly control. R is something that you can only indirectly control, and the magnitude of your control, etc., etc., you can only figure out when the experiment is over, when the entire time series is in your hand, and then you fit a model, and from that you get your R, or that you get your K. Okay, so there is no experimental access to the system parameters, which makes much of the modeling that has been done in the chaos control literature, or for that matter in the control literature, not applicable or impossible to apply, let's say, in the biological systems. The third thing that happens is that many of these methods, they I'm sorry, many of these experiments, or population dynamics data, they are short time series and they are noisy time series. And as we were discussing, given that kind of time series, it is very difficult to distinguish between something which is chaotic and something that is, let's say, a reasonably regular dynamics with a little bit of noise around that. So under such a situation, one can ask the philosophical question that does it really matter whether it is chaos versus non-chaos? Okay, given that for all practical purposes, visually the two dynamics are looking extremely similar to each other. And finally, much of the methods that have been proposed, they are in the context of noiseless systems. Okay, and any biological system is inherently noisy, which makes the utility of those methods in controlling them rather shaky, okay, rather problematic. And another issue that happens with many biological populations is that they can undergo extinctions, local extinctions. And as we are going to show, the moment extinctions come into the picture, the utility of these methods are greatly altered. Okay, many of the methods basically fail to work the way they are supposed to. So faced with all these realities, what does one do? So the ecologists obviously are aware of all these problems. So the theoretical ecologists, they have come up with methods in which, instead of focusing on the system parameters, the analogs of the R's and the K's, you actually focus on the state variable. You focus on that thing to which you have an access as an experimenter. So for example, if you guys remember the method that we discussed in the last class, where we said that every generation we are going to add a constant number to the population. So technically speaking, that is called pinning. So this kind of a pinning is something that is being done with the individuals, and therefore this is something that you can experimentally play with. So the methods that ecologists came up with, most of them were of this type, where you state variables directly, and then instead of concentrating on chaos versus not chaos, you concentrate on things that are more biologically relevant for the dynamics, things that are more unambiguously measurable. Okay, And in this context, the kinds of stability that people have concentrated on are what are known as constancy and persistence. I mean, these are just examples. They have also concentrated on other kinds of stability. I'll come to this context of stability in a few minutes in a big way. So, if you look at the literature, then one of the most promising candidates was the one candidate that we discussed in last class, which is, as I said, called pinning, which essentially is constant immigration. And although, as you guys, some of you saw with your own simulations, that it seems to work under theoretical conditions, the question was, does it also work in real experiments? So during my PhD, I did one piece of work where we actually ended up taking spatially structured populations. So where you have multiple populations which are connected with each other, connected to each other by migration. So we wanted to see if one can stabilize the dynamics of such populations using this kind of constant pinning. And we ended up seeing that, as the title says, that it didn't really work, although all simulations suggested that it should. And we found out that the reason for which it is not working is because of the extinctions. So if extinctions of local populations is something that you just cannot avoid in almost any spatially structured population, and that completely kills the way pinning works. So when I joined here in 2007, a few days after that, this girl came to me for doing her master's thesis. And those days the lab was just starting, so we said, okay, let's do something crazy. So what we did was, we set ourselves a problem that since pinning had not worked, we wanted to find a different method which could stabilize the dynamics of populations. And since we were doing it, we said, okay, let's do both, spatially unstructured, that's what I call spatially homogeneous, and spatially structured ones. Okay. And we wanted to come up with a method which should be implementable in a real population, so not a mere theoretical stuff. It should be robust to noise and extinction. And this is very important. See, many methods proposed in the literature are shown to be effective in parameter zones which are very, very difficult to be biologically realistic. Okay, so we wanted this to be applicable in a parameter range which was biologically meaningful. And then once we created this method, we thought that we should run an experiment and empirically validate it as well. So after looking at the literature, we looked at various classes of methods, and we figured out that from an experimental point of view, the class of methods which makes the most sense is what is known as limiter control. So limiter control says that essentially do not allow the population size to go either above or below some kind of a threshold. Okay. Now, obviously, when you set your brief like that, then the question becomes, how does one identify this threshold? Okay, so different populations are going to have different equilibrium population sizes, the equivalent of K. They will probably have different values of R. So will the same size work for all of them? And the answer is obviously no. Okay, so if you guys take your constant immigration, the pinning thing, and you do some experiments and numerical experiments on that, you will figure out that as you change your K, the value of C for which you obtain different dynamics will keep on changing. So if that be the case, then how does one set the threshold for different populations? How can one come up with a method which is robust to this problem? And the very obvious solution to that seems to be that make that control, whatever magnitude by which you are going to perturb, dependent on the population size itself. So after some amount of searching around and etc., this is the method that we came up with. So what we said was that this is the mathematical equation, but biologically speaking, what it means is that whatever happens, don't allow the population size to go below a fraction C of the size of its previous generation. So suppose we set C at 30%, 0.3. So if the population size in the previous generation is 100, then this generation I will not allow the population size to go below 30. If it is 30 or above, I'll not do anything. If it is below 30, I'll bring it up to 30. That's it. Okay. Now, as you can see, if I can set C equal to 1, then I'm saying that each generation size will be brought to a particular value, which is trivially stable. Okay, but that is something that is not of any interest. So we are actually interested in much lower values of C, and we want to see whether those lower values end up getting stability or not. Okay, so we decided to study. By the way, people, I can't see your questions when I'm in this presentation mode. So if anybody has any question, you have to speak it out. Okay. So yeah, so we used the same Ricker model that we were talking about for modeling the dynamics. Why did we use the Ricker model? There were three very excellent reasons for it. Number one, as you can see, that this model is a very generic model. It has just two parameters, this R and K, and none of them are any species specific parameters. None of them encode some kind of phenomena which only happens for some species and not for other species. Okay, so in that, if we end up getting something using, getting some predictions using this model, then one can at least hope that this is something which will be generalizable. More critically, this particular model has been derived analytically from first principles based on some very relevant biological assumptions. Essentially, the two biological assumptions that it comes from are A, the populations, the individuals in the populations are distributed randomly in the space, and B, they have a particular kind of competition known as scramble competition. Now, what is scramble competition? We'll deal with in the next topic when we talk about competition, but it's a very common form of competition that is present across vast parts of the animal kingdom, and therefore it's not at all surprising that this particular model is empirically, it has been shown that it is capable of explaining the dynamics across a very large wide. So basically this model has been applied for bacteria, it has been applied for fungi, it has been greatly applied for insects, it has been applied for several other invertebrates, it has been applied for fishes. Okay, so it's in the context of fishes that this became famous to begin with. So fishes and yeah, so if you look at this, bacteria all the way to fishes, this between itself probably takes care of 95 to 99% of the dynamics out there. Okay, and 95 to 99% of the species out there, and therefore the dynamics. So it is one of the most, it's one of the most widely used and widely relevant model of population dynamics in the literature. Yeah, somebody had something. Okay.
Yeah. Can you hear me?
Yeah.
Didn't you tell us that for bacteria, for things like bacteria, we tend to use continuous models?
Yes, but you can also grow bacteria under discrete situations in which you can use this one. So this is a discrete model, na? So you can also grow bacteria as discrete populations.
Acha.
Then this can, then this will work. As a matter of fact, okay, if you look at the time series of this thing, for R values sufficiently low, it exactly does sigmoid dynamics, right? Do you guys remember that part? Yeah. So, so that
it doesn't
also
oscillate at all.
Exactly. So that shows that under those conditions, this will directly work. Okay. Anyway, but what I'm talking about are discrete conditions. So you can also have bacterial model under discrete dynamics. Anything else?
No, sir.
Okay.
Not from me, at least.
Okay, okay, cool. So this is why we use the Ricker model. Okay, now we come to the context of stability. So if you guys remember, when we were doing that, all those bifurcation maps, etc., etc., we said that when a dynamics is, let's say, a single point equilibrium, then that is more stable than, let's say, a 2-point limit cycle. A 2-point limit cycle implicitly is more stable than a 4-point limit cycle, and chaos is the least stable. Okay, so the complexity of the dynamics was what we were taking as a very crude measure of stability. And even there we said that, in the presence of stochasticity, even that goes out of the window. So how does one quantify stability? Now, in mathematics and therefore much of physics, stability is rather unequivocally defined. Okay, so mostly people see that, if you are slightly perturbing something from the equilibrium, then how quickly it comes back to the equilibrium state, and how quickly it does so and how reliably it does so is a measure of how stable the whole thing is. The faster it comes back, the more stable it is. Okay. However, it turns out that in ecology, the concept of stability is hopelessly muddled. So there was a 1997 paper in which they actually did an inventory of how many definitions and how many concepts of stability exist just in the field of ecology, and they showed that there are 163 definitions of stability related to 70 independent concepts. Okay, so that's how muddled stability is in ecology. And therefore what these guys did was they did a very nice essentially, a taxonomy of stability concepts, and ended up showing that most of these can be actually grouped under 5 or 6 big concepts. Okay, and out of these, they explicitly pointed out that 3 concepts, what are known as constancy, persistence, and resilience, these 3 are the most relevant for the various biological theories that are around. So, what are these things? So, constancy, stability, is essentially related to the amplitude of fluctuation in the time series. So, if you have 2 time series and one has a much larger amplitude of fluctuation in population size over time, then that is supposed to have lower constancy stability compared to one which has a smaller amplitude of fluctuation. So, uh, let's see if I can show it to you guys. Yeah, so suppose, oh, so suppose there is one population which is doing this, and there is another population which is doing this. Okay, so you have time on x-axis and you have population size on y-axis. So then the first one is going to be called, this one is going to be called less constancy stable than this one. Okay, now how do you measure it? There are many ways of measuring it. The one that we ended up using is what is known as the fluctuation index. What it essentially does is it takes these lengths, okay, up lengths and down lengths, sums them all up, and then essentially takes an average over the entire thing. Okay, and scales it by the population size, the average population size. So by doing so, this becomes a dimensionless quantity, in which case it is very easy to compare it across things. Okay, so this is constancy stability. The other kind of stability that is very important biologically is what is known as persistence. So if you remember, I was saying that whenever you have a spatially structured population, then the local populations, they end up going to extinction all the time, and therefore one measure of stability can be how long they take, or within a given time span, what is the probability of their going extinct. So the idea is the ones that have a higher probability or a higher frequency of going extinct are less persistent stable compared to one that has a lower probability. Okay, so you can see that these are all things which are measurable much easily, and much more unequivocally they can be measured, and they can be measured even if you have relatively short time series. You don't need long time series, you don't need fancy mathematical methods to use this. So these were the two things that we ended up choosing, and we did our experiments using Drosophila melanogaster. So this is the common fruit fly. Did you guys handle this in your first year? Typically there is an experiment on Drosophila.
Yeah, the
We got to see it.
observation. Yeah, we are allowed to just see it.
Yeah, it was some
Okay.
model organisms thing. So.
Okay, okay. So anyway, so you guys know how small they are and how they look and so on and so forth, right? So what we did was we took 3 treatments, and this one was C equal to 0, which is the control, C equal to 0.25, which we call lower ALC, and C equal to 0.4, which we call high ALC. So what C equal to 0.25 means is that if the population size in a given generation is less than 25% of the population size of the previous generation, then we'll bring it up to 25% of the previous. But if it is higher, then we will not touch it. Okay. Similarly for 40%. So this is what a population looks like. So it's a single vial culture, and each generation we counted how many flies are there just before breeding. We applied the ALC based on whatever value we have chosen, and then after doing this we recorded the census size. We allowed them to breed for the next generation. The parents were discarded and the eggs formed the eggs essentially led to the next generation. So we kept on doing this for 13 generations, 8 replicate populations per treatment. So first let's look at whatever we have here on the right side.
Sir.
Bolo?
Yeah, so when you say that you have to pull up the population, bring up the population, do you introduce new individuals?
Yes. So basically we had backup vials, and in order to make up the number, we took some flies from the backup vials and we introduced them to make up the number.
Okay, sir. Thanks.
Okay. So what you see over here on the right side, these are the simulation data. Okay. So what you have here on x-axis are different values of C, that ALC proportion, and whatever you have here is the population fluctuation index, the formula that I showed you guys, plus minus standard error. So as you can see that the population, the simulations are predicting that as we increase the value of C, so this is 0, this is 0.25, and this is 0.4. So as we increase the values of C, the fluctuation index is supposed to fall. Okay, and now you can see why we chose these two values, 0.25 and 0.4, because our simulations suggested that we should be getting a very nice gradation between them. Okay, and this is the experimental result. You can see that we get a very nice gradation again, and roughly speaking, I mean, I won't put too much to it, actual values of Fi that we are getting are roughly in the same zone as these ones. Okay. So this suggested that ALC significantly enhanced the constancy stability. So lowering of Fi is equal to enhancing constancy stability in the experimental populations.
So why is the error more for the control case and then it reduces? We see the error bars.
Experimental data, very hard to say much. One thing that is happening over here is that extinctions are happening more over here. Okay, see, one of the things that ALC does is it kind of makes sure that there are no extinctions, because you are always pulling the population up. Okay, now, yeah, so if you look at that constant immigration wala thing, you realize that when you increase the stuff, when you what is meant by increasing the stuff, when you increase when you introduce some individuals from outside, then typically for some low values you actually end up reducing the fluctuation index. Okay, you end up reducing the amplitude of fluctuation, and since that is not there in this particular case, you are expected to get a slightly wider variation. Is this making sense?
Yes, sir.
Yeah, we can discuss this later. It's a nice technical point. we'll have to discuss this in the context of the return map. Okay.
So
Okay.
हाँ, बोला?
the one more thing, when we were talking about chaos, we had talked about how
Mm.
similar initial conditions can lead to very different behavior, very different dynamics. Is there a
Yes, yes.
shouldn't there be a side of stability or some definition of stability that also takes into account how stable the dynamics is, even if you perturb the initial conditions? Because that
See,
will also matter a lot, right?
Yes, but the problem is that that thing is implicitly assuming that you have a not noisy system. Because if you have a noisy system, then
Right.
even if you start from exactly the same thing, you know there is going to be divergence.
Okay, yeah.
So which is why for these kind of experimental systems, the sensitive dependence to initial condition thing has no meaning.
Acha.
I mean, you can't attain it anyway.
Okay.
The other problem is that biologically speaking, what is the closest that you can take them? The closest you can take them is one individual.
Right, right.
Which is already pretty far as far as SIT
Okay.
is concerned.
Okay.
So that's why in these contexts we don't even talk about it.
Right.
Yeah. So
So,
this was about concept.
sorry, what is the P here? P less than 1 by 1000.
Oh, acha, acha. Okay. So see, the point is that whenever this is experimental data, right? So whenever we are doing this, we have to show that this point is different from this point and statistically different from this point and so on. So we do what is known as analysis of variance to show that whatever you are getting over here, this difference is more than what you expect by mere randomness or mere variation around the data. So that is what is this is the result of the statistical analysis. Okay.
Okay, thank you.
Anything else? Okay. And this is the reason for which I typically don't want to do this. We have 39 minutes into it and I have not even finished the first one. Okay, anyway. So what happens to persistence? Remember, persistence is frequency of extinctions. So turns out that ALC does have an effect on persistence stability, but the effect is essentially when you go from control to lower or higher. Okay, these two are not different amongst each other, and frankly speaking, if you do a statistical analysis, this result is not significant, which essentially means that although we are able to see something, you can't say much given how much of noise is around the extinction values to begin with. In a nutshell, this adaptive limiter control, it was able to stabilize the dynamics of spatially unstructured populations. Why are we calling them spatially unstructured? Remember, we took single vials and everything is happening inside the vial itself. Okay, so there is no spatial structure. Now, what happens if we go to a meta population or a spatially structured population? So what is a meta population? So when you have subpopulations, local populations, which are independent enough so that they have their own dynamics, and yet they exchange a few individuals, some number of individuals through migration, then this entire ensemble that you have, this entire ensemble is what is known as a meta population or a spatially structured population. Now, one of the well-known theoretical results and an experimental result from my own study previously was that when you change the migration rate across these subpopulations, then that can change the amplitude of fluctuation at the metapopulation level. Okay, so that's what I mean over here, saying migration rates can affect metapopulation constancy. So when you are studying the effect of ALC over here, you also need to factor in migration rate as a variable. Okay, because it is entirely possible that ALC can have one effect at one migration rate and another effect at a different migration rate. So what we did was we had two patch metapopulations. So what basically means is that we had two vials and we counted the number of flies in each one of them, and each generation we also made the flies migrate, some fraction of flies migrate from here to here and vice versa. So we took two rates of migration, 10% and 30%. There is historical reason for this, because experimentally we had shown that when the migration rate is 10%, then the metapopulation becomes very stable. The amplitude of fluctuation in the population size becomes low. Whereas when the migration rate is 30%, then the amplitude of fluctuation in population size at the metapopulation level becomes very, very high. So this 10% is a stable metapopulation. This is a relatively unstable metapopulation. And what we did was we applied ALC to one of the populations, and the idea was that this application in one, is it able to, or is it sufficient for taking care of the whole ensemble or the whole metapopulation. Now please remember, we have already shown that if you have one single population, especially unstructured, then ALC will bring the FI down, not a problem. Therefore, if we ended up applying it to two of them, then individually each one of them, their fluctuations would have come down, and therefore the ensemble's fluctuation would have also come down, okay, which is not an interesting result. Therefore, that is the reason for which we ended up applying the ALC just to one vial and not to both. So again, here is the simulation. So in this case, this upper one is the 30% migration, and you can see that you expect the so you see here on x-axis, you can see that you let me just finish this. So you can see that the fluctuation index here is always expected to be greater than the fluctuation index for the 10% 10% migration wala metapopulation. Yeah, tell me.
The migration rate is the same in both directions, okay?
The migration rate is symmetric.
The migration.
Yes.
Okay.
Yeah. You can also do with asymmetric. That just makes the matter more complicated, because if you are doing asymmetric, then in principle the effect of applying it in this one is going to be different from effect of applying it in this one. So we
that's why I asked. That's why I asked about that.
No, we kept it symmetric. Okay. So
experimentally, how is the migration rate controlled?
So you actually count how many flies are there, and then you take 10% of that and push it to the other vial and vice versa, or 30%. Yeah.
Okay,
so very tightly controlled. See, this is the reason for which we can actually talk about the migration rate. Otherwise, if you simply connect them using some pipes, then you cannot talk about the migration rate, because the flies will migrate to whatever fraction they want. Okay. So as you can see, the simulations are suggesting that in the 30% migration case, you expect a nice fall over here, and you don't expect too much of a difference over here. Now you can see that the experimental results are showing exactly the same thing. There is going to be a big nice fall over here, but you don't expect too much of a difference over here, and as a matter of fact, this difference is not statistically significant. Here you expect a nice fall over here, but not actually a slight increase over here, and overall the magnitude of difference that you expect over here is not that large, because even the simulations are suggesting that the control is going to have a rather big error bar around it. So essentially we got something which is roughly in the same direction. We saw all is not much, and again there is not much of a difference over here. So essentially, if you synthesize all of it in one place, what this is saying is that when you have high migration rate, then ALC is going to do something, but whether you are having a higher rate of ALC or a lower rate of ALC is not going to matter too much. Okay, which in some sense, this result is not very surprising, because it's only when the migration rate is high that the population is unstable, and you can only stabilize an unstable population. If the population is pretty stable to begin with, as you find over here, then it is hard to stabilize it further, because there is a residual amount of noise which will always be there. You can't do much about it. Okay. So anyway, and so essentially that it enhances the constancy of the unstable metapopulation, this one, the 30% one, but does not affect the stable one. And if you look at persistence, then it turns out that under both circumstances, the metapopulation level persistence actually becomes sorry, not metapopulation, subpopulation level persistence actually becomes much better, but again no difference between the ALC 0.25 and ALC 0.4, suggesting that this is a good method for promoting persistence stability, right? So just to summarize, that this particular method is a potent mechanism for enhancing constancy and persistence in real biological populations and metapopulations. And since our empirical results are being supported on a host of non-Drosophila specific models, so I showed you the results from only one model, the Ricker model. We also did it on other models, and in all the models, this particular whatever trends we are getting, the trends were pretty much the same. Therefore, we ended up suggesting that this is something that is likely to be very broadly applicable. So we ended up publishing this paper, and we said that this is the first control method which was empirically shown to work for a real spatially structured population. And this method is nice because this is also the first method which reduces both the fluctuations as well as the extinction probability. So enhances both constancy and persistence. Okay. So then obviously the next big question was, how much can you generalize? What happens if you have other rates of migration? What happens if instead of two subpopulations you have more subpopulations? What happens if you change the values of k? What happens if extinction rates are changed? What happens if you have different ways of putting in extinctions and so on? So any theoretical study, these kind of questions always arise. So we did a very, very, very extensive theoretical stuff on this, and essentially we ended up showing that this method, adaptive limited control, is going to work in under all these circumstances. So till this point, whatever we were doing, we were essentially talking through simulations, and we were pretty confident that our thing is very generalizable, because as I said, we were able to apply it in a large number of models. Our experimental system was showing that this was doing very well, and so on and so forth. However, all said and done, we didn't really have a analytical solution for this. Analytically, we could not say why ALC was working. So that's when these two gentlemen entered into the picture. So this is Daniel Franco Leis. So as you can see, he is a mathematician from Spain, and this is Frank Hilke. He is he's not a mathematician. I think he is okay, I forget what his qualification is, but he was at the School of Mathematics and Computer Science in Germany. So these guys, they picked up our paper, and they actually mathematically, analytically established why is it that ALC was able to attain stability in the single populations. Okay, so this is a completely unreadable paper, at least for normal biologists like me. It has got theorems and lemmas and God knows what, but at the end of it, it nicely says that this is why ALC works. And in the process, what they also ended up doing was that they ended up many other things that we had just put out as experimental observations, they ended up putting all of that on very firm mathematical footing. Okay, they gave the analytical reasons for which those results are the way they are. And then the last 4 or 5 years, these guys have actually given several other variants on this adaptive limiter concept. Okay, so I think there was a paper as late as 2019, which I have not included over here. So till now, okay, what we have seen is that this adaptive limiter control method, it enhances both constancy and persistence, works for both single as well as specially structured populations, spatially structured, and it is actually robust to several kinds of biological realities. And most importantly, it has solid mathematical explanation for why it works. And between those guys and us, we have made ALC to be one of the most well-studied control algorithms in the whole population dynamics literature. And with this, we come to the absolutely million-dollar question. So what? Okay, because see, we have, what I talked to you about was one control method. Okay, and I gave you guys a nice account of how it has been empirically verified and how mathematically supported, so on and so forth. However, the problem is that there are so many other control methods in the literature, and all these methods, they have been proposed in the context of different models. They have been checked out in the context of different model parameter ranges, so different values of R, different values of K, and so on. They have been talked about in different contexts of stability. As I said, there are so many ways in which people have looked at stability. And the issue is that if you put in large enough control parameter values, then almost all these control methods can induce almost any level of almost any kind of stability. Okay, so suppose you are a person, you are a, let's say, a manager who wants to actually employ a stability method, or actually employ a control method, then which method are you going to choose? For that, you need to compare across all these methods, and such a comparison essentially just didn't exist. So what would one need to do for doing such a comparison? For doing such a comparison, one would first need to fix a model. Okay, it has to be done in the context of a given model. You can't compare Ricker with Hassel with logistic with something else. And then for that model, you have to fix a particular target. Okay, it can be anything, but it has to be a fixed target, say a 50% reduction in FI. And then for each method, figure out the parameter values at which that level of stability is going to be attained. And then you have to figure out the efficiencies of these various control methods for those parameter values using other kinds of criteria. So things like, what will it cost to have this method employed? Or, whenever you are changing the population size, you are also playing around with the amount of genetic variation that is in the population, which in turn will have effect in terms of how the population evolves, so the evolutionary trajectory. So that is something that is caught by what is known as the effective population size. Okay, so how does the effective population size of the method of the population gets affected by whichever method you are using? And then essentially you end up integrating the performances of all these methods using some kind of a composite index, and only when you have done all these steps, now can you directly compare one method against another, apple to apple comparison as opposed to apple to orange comparison, and then say this is a good method and this is not so good a method. Okay, so where are we? Oh god, we are 10 minutes above time anyway. So what I'm going to do is I'm going to stop right now, and in the next class we are going to look at how all these things work. Okay, what happens when you do all these things and what kind of answers you end up getting. Okay. Oh god, there are 30 chats. Okay. Oh, so the number of people are reducing. We have only 90 people today. Cool. Okay, so since it's 55 minutes already, what I'll do is I'll stop the recording. So the class will come to a close, and then if people want to hang back, I'll hang back for a few minutes and answer a few questions. Okay, so officially the class has now ended. Okay, it is saving in my drive, so